Hello! So today, we're going to make the 3D model of the bacterial cell. Let's start! So these are the materials that we're gonna be using. So the first step is get your carton and your cutting tool. Then, get your pencil and a ruler. As you can see here, I'm marking my desired shape for my capsule. After marking your desired shape, you can now cut your capsule. After cutting my capsule shapes, I am now assembling them. And tada! This is my capsule. I cut another piece of cardboard for my cell wall. Then I just mark it half an inch. As you can see, I already covered my capsule color blue, and then I added my plasma membrane, which is the red one, and then I added my cytoplasm, which is the mint green one. The next thing I added is my cell wall. It's color yellow already because I did it off camera. Then I'm gonna be using the yarn to make the DNA. Then I cut smaller pieces of the yarn to make the plasmid. After adding the plasmids, I am now adding my ribosomes. After adding my ribosomes, I am now adding my pili or my pilus. So I did it off camera and this is how it turned out. Now we're gonna add the tail-like part, which is the flagell. Then I added numbers for the labels. And this is the finished product. So let's talk about the parts of the cell. The cytoplasm is a jelly-like substance between the nucleus and the cell membrane. The next one is the plasma membrane, also known as the cell membrane or the cytoplasmic membrane. It's a biological membrane that separates the interior of a cell from its outside environment. The primary function of the plasma membrane is to protect the cell from its surroundings. Then the cell wall. It provides the cell with both structural support and protection. It also acts as the filtering mechanism. Pili or the pili, also known as the fimbriae. The function of this is to attach the cells to the surface. The DNA is a complex molecule that contains all the information necessary to build and maintain an organism. The next one is the ribosomes. Ribosomes are the sites in a cell in which protein synthesis takes place. A plasmid is a small, circular, double-stranded DNA molecule that is distinct from a cell chromosal DNA. The capsule contains water which protects the bacteria against desiccation. The capsules also protect the bacterial cell from ingestion and destruction by the white blood cells. And the last one is the flagell or the flagellum. Flagellum is primarily a motility organelle that enables movement and chemotaxis.